The first speaker here, that's Richard Cardoso Da Silva. He's from Sao Paulo, Brazil. Did research in many different places. He's been in Brazil itself, in New Zealand, Max Planck in Germany, and more recently with Tunkai Bobek's group in Zurich, and then moved over to Utrecht, is my understanding. Working on the proteome, but also the epiproteome to understand dynamic cellular processes. So looking forward to your talk. Thank you.
Thank you very much. Well, thanks everyone. Good morning. Thanks for the opportunity to, I think I need to talk on the microphone, to present my work today here. I'm very excited. Today, my talk, we're going to delve a little bit into chromatin biology, essentially trying to understand how can we use engineered probes to understand chromatin regulation. Yeah, so I'm just going to give a brief introduction about chromatin, but as you know, our the chromatin is highly organized in the nucleus. Our DNA is wrapped up essentially around histone proteins, and these histones make the fundamental unit of nucleosomes. What is important to emphasize here is that, as you know, your chromatin is more an open chromatin state that transcription is prominently active, whereas heterochromatin is densely packed and transcription is silent. So what is the contribution of chromatin to genome function? What we know is that how can a single cell with a single genome give rise to many different cell types with specialized sets of gene expression? That's a fundamental and long-standing question in biology. We know that just encoded proteins itself are not sufficient for that sort of complexity that we see in these different cell lines. But we know that most of this complexity comes from regulation of histones and chromatin. And this sort of complexity comes mostly from this diverse array of covalent post-translation modifications on histones, but also on DNA, in the case of DNA methylation. So these epigenetic landscapes with different histone PTMs and DNA methylation, for example, is essentially read and interpreted by a group of proteins called reader proteins. These reader proteins are a diverse range of different proteins that can read and interpret these different moieties, either on histones and on DNA. These reader proteins are actually modules that are part of many different proteins, these reader proteins. So how do they work is that they usually recruit many proteins to the chromatin, and this recruitment is pivotal for the activation and repression and to modulate gene expression, and therefore all this complexity gives rise to these many specialized types with different sets of gene expression that we know. In our group, we previously used an approach which we named chromatin Lego, but essentially what we do is that we use these domains that have affinity towards chromatin modifications as modules to probe different mechanisms in the cells and different pathways. So, for example, how we do that is that we assemble these modules into cell line by using recombinase-mediated exchange. So what we do is that we have a cell line that we can essentially clone many of these different flavors of readers domains with affinities towards different histone modifications. And we can use these cell lines, for example, to probe repressive chromatin in the case of H3K9 trimethylation, facultative heterochromatin, active promoters, bivalent promoters by use combinatorial readers, and also the DNA methylation. And our group have been successfully employing these readers, for example, to assess dynamics of chromatins in live cells when you fuse those readers with GFP. And this allows us, for example, in this example, to track facultative chromatin in live cells so we can understand dynamics over the cell cycle. We can also use these ECRs expressed in the cells to look at genome-wide binding aspects, for example. But finally, what we have successfully using these ECRs for is to assess the proteome composition around these specific marks. So you can actually ask okay, what are the proteins around different chromatin modifications, for example, in heterochromatin and in chromatin, and what are the regulators that are recruited with the use of these readers. So we believe that this is an important toolbox, for example, for us to understand basic and fundamental aspects of chromatin biology. We know that there are different moieties that can be explored, either in acetylation, phosphorylation, or bucosylation. So there's a very vast array of different moieties to be explored. And for that, what we've done, it's a tour de force for many different people in the lab along the years. We created a big panel of cell lines expressing different readers with different flavors towards different chromatin modifications. We have, and we are still currently characterizing many of these readers and their affinities toward different chromatin modifications, to use this as some sort of affinity tool or affinity reagent to ask questions pertaining chromatin biology. I am mostly interested, for example, and that's what I'm going to guide you now on this talk, is in phosphorylation in a histone variant called H2AX. So H2AX is a histone variant that is releases plastic-4 damage and cells coming from the environment. I will just give an introduction about this. So whereas we have the unperturbed chromatin and all these regulatory processes that are taking place in there. But upon a DNA double-strand break, what happens is that this DNA break, which can be, for example, from exogenous sources, many different drugs, ionized radiation, UV, or also they can be from essentially endogenous sources. For example, lots of double-strand breaks arise during DNA replication. So upon double-strand break, what happens is that this histone variant is phosphorylated by specialized kinases, as you see here. And the name of the phosphorylated version of this histone variant is called gamma H2AX. Gamma H2AX is a very important and well-characterized hallmark of DNA damage, as you might have seen before. So this histone gamma H2AX is actually serves as a signaling platform for the recruitment of many proteins, not only to deal with the DNA repair, which is also very well characterized, but also to allow the chromatin to be repaired, but also to allow the chromatin to fold and to change and to adapt to the new scenario, which is a damaged chromatin. So what is happening is that once you have a phosphorylation, there is this folk organization of the chromatin, and the folk organization of the chromatin allow us to identify DNA damage because this forms a foci in the cell, and this is a very well-known and characterized marker for double-strand breaks. And it can indeed be identified via microscopy with antibodies against gamma H2AX. Thinking about that, we screened and characterized readers, essentially phospho-binding modules that would have affinity towards this gamma H2AX, toward this phosphorylation, that would allow us to track these proteins in living cells. So what I did, I looked for many candidates, which I'm not going to show here due to problems with time, but essentially I found that a protein called MCPH1, when fused with VEGFP, is essentially the BRCT domain of this protein. It does colocalize and is tethered to double-strand break sites, as you can see here. And indeed, when we stably integrate this reader in cells, it does fully colocalize with gamma H2AX in living cells. We have already characterized this probe as a bona fide probe because it does not affect with global repair and the cells are viable. So this actually is a very nice tool so we can actually track double-strand breaks in living cells. In contrast, when you do express only a nuclear VEGFP, as you expect, you see this sort of a diffuse signal. And when we add point mutations in the binding, the phosphor pocket of this reader, the signal is pretty much gone. So the question here is that all these proteins that are recruited to these phosphorylation sites, most of them have been very well characterized in the DNA damage field because lots of these proteins are DNA damage repair proteins. But the question is that we want to understand and find novel proteins and novel hits that would allow us to further advance the field of the DNA damage. So that's why we decided to use this probe that has very good specificity towards gamma-H2AX for proximity bioaccumulation studies. So what we did is that we stably expressed this same construct now in fusion with turbo-ID, and then we treat the cells with different genotoxic agents. In this case, I'm showing an example of etoposide. And then what happens is, as expected, double-strand break is triggered, phosphorylation of gamma-H2AX. We know and we have observed that the reader efficiently tethers the turbo ligase to these sites. So we can ask ourselves, upon treatment of cells with biotin, that we can actually record all these proteins around a break. And this gives us the chance to record all these proteins and essentially to enrich these proteins by pull down and then detect these proteins by mass spectrometry. So what we found, as you can see here in this volcano plot, is that we get highly enrichment upon treatment with etoposide. And you can see here that we do get a bunch of the proteins that are expected to be recruited to these sites upon double-strand breaks. And I will just give an example of some of them. For example, when we look at some of the high confident proteins, we get, for example, H2AX, which is the histone that is being phosphorylated. And interestingly, we also get the protein ATM, which is the kinase that phosphorylate H2AX. We also get many other DNA repair protein that are well characterized, like 53BP1 and RAD51. I will spare you of the agony of going through all these proteins here, but just want to emphasize that we also get many other chromatin regulators, erasers, chromatin remodelers, and these are basically the ones that we are interested in characterize the additional proteins. And as expected, DNA double-strand break proteins are significantly enriched in our data set. When we treat the cells with DMSO, as expected, we only capture proteins that reflect mostly the basal double-strand breaks that are endogenous, coming from regular processes in the cell. So the next question was just to ask ourselves whether we can capture different conditions, whether the genotoxic agents allow us to identify specific hits with this data set. And with that, I show mostly a network where I synthesize here all the proteins that are found in these different conditions. And you can see here that, for example, when we look at camptothecin, which is a drug that triggers a specific break via a specific mechanism by inhibiting topoisomerase 1, that top 1 is only identified in camptothecin conditions, whereas in other conditions, we do not identify. So that's the main idea of this project, is really to capture genotoxic specific proteins that allow us to say, okay, if we treat the cells with UV, what are the complexes and what are the complexes that we get and we lose during the particular processes. And then I want to emphasize that we also capture proteins involved in spindle pause and kinesin complexes and cohesion complex, which give us a very nice and broad idea of what are the complexes that we can actually discriminate with this approach. And essentially, what we have found so far is that we do get a bunch of proteins that are expected, which is good. It shows that the approach is very accurate to capture proteins in double-strand break repair and pathways. But we are really interested in proteins that are unreported and factors that are still not known to be part of the DNA damage response. So we can actually further and advance our knowledge about these particular processes. And obviously this opens up possibilities to identify unreported factors and proteins that can be valuable as resources for the field, mostly connected to chromatin. I kind of lost track of time, actually. 3 more minutes, yeah. Essentially, I want just to sum up some of these aspects here. It's important to mention that we have uncovered and we have screened for a probe that is an unbiased probe that is not really affected by many other aspects of the cell. This unbiased probe is very specific to gamma-H2AX and is very useful, for example, to track dynamics if you want to use microscopy with your favorite drug, with your favorite genotoxic agent, or a particular mutation that you want to test. And we can track these breaks over a full cell cycle, for example. And I'm showing here that we can actually use this reader to track the proteomic composition around double-strand breaks with different conditions. We've tested a bunch of them, UV, cisplatin, so on and so forth. But this opens up for the possibility of identify many other proteins, because there are still essentially a lot of genotoxic agents that are available. So I think that's the next aim, is really to expand this with different drugs and actually assess quantitatively all the proteins that are recruited to these sites. And also we would be interested in characterize and map the proteome composition in different cell types to actually see what is the pattern of double-strand break repair and chromatin associated regulators to these double-strand break sites. This is also really interesting, and something that we wanted to focus is really we can target the breaks through different chromatin regions, for example, and we can capture the double-strand break in specific parts of the chromosome and in the chromatin. For example, by targeting breaks in the centromere, we can actually get the proteome composition of breaks only taking place in the centromere, whereas if we track the telomere, we can see what are the protein composition around telomeres as well. And finally, to look and use this probe as a tool to understand fundamental processes in other model organisms. And in fact, we've been using successfully this probe to track programmed meiotic double-strand breaks in C. elegans. And with that, well, I'd like to thank everyone involved in this project, especially everyone in genome biology and epigenetics. It's been very contribution from many people in the group. I'd like to particularly thank Kelly Stacker and Christina for all the turbo-ID and proteomics, and also other collaborators. If you're interested, there's a bioarchive with part of this data that has been submitted, and we are still working on the manuscripts. And also I thank all the funding. And I thank you all for your attention, and if you have any questions.
Thank you very much for a very, very interesting talk. We have time for two more. We have 2 minutes time for questions.
Thank you for the very nice talk. So I'm not very familiar with this topic, but I was just curious how different reagents can cause different type of proteome around these breaks, because DNA break is DNA break, to my knowledge. So yeah, that was interesting for me.
Yeah, essentially the break, the path into a particular break will have some sort of intricacies in between. So for example, this drug that I mentioned, a toposide, will trap a particular topoisomerase, and the whole pathway to the break will trigger different repair proteins. So this is actually very well characterized. Or if you treat the cells with UV, essentially initially the cells they might have single-strand breaks, and then single-strand breaks might give rise to double-strand breaks, and so on and so forth.
Fantastic work. So could these different signatures of associated proteins have some predictive value, whether there can be a response or exceptional good response to these treatments? Because I can imagine if there's alterations already at baseline that the drug may or may not work as good. Have you explored that?
No, not yet, but that's actually a good point, because I think the idea now as a proof of concept is to get the system to work and to test these different drugs. But we know that we can discriminate essentially on a PCA, for example, group of proteins that are UV dependent with group of proteins that are toposide dependent and cisplatin dependent, for example, and a core bunch of other proteins that are very general. I think in the future maybe we could actually explore this, but we haven't explored much. I think there might be the possibility to also increase the identification of proteins by, for example, going for DIA instead of DDA and having more increased number of hits that can actually allow us to do some sort of quantitative towards that direction.
All right, thank you very much.
Thank you very much.
I would like to welcome